Okay. So the lab that you are going to be doing in class today is called A Close Look at Flowers. And we are going to be going over the different parts of a flower today. You are actually going to get to take a look at a real flower, and you're going to dissect the different parts. And you're going to be labeling and figuring out what roles the different parts play. So your materials that you are going to need is a paper towel, which I have laying right up here, a hand lens to look at anything up close, a large flower, which I will provide for you, a scalpel or a razor blade, which I will be handling because it's very sharp, and then also a roller to measure the different parts of the flower. So it's pretty simple. We're going to be working from the outside of the flower in. So the first part of your procedure is going to be to measure and count and make observations of the petals. Alright, so you're going to come over to the vase and get a flower. And then all you need is just the stem up. So break it off. And then, like I said, we're going to work from the outside of the flower in. So put the paper towel down on your area so that you don't make a mess on the table. And you're going to make your first observations of your flower, and you will be putting these observations on the lines next to the bullet points. You will then have to measure the petals on the flower, so you will just simply remove all of the petals. And then take your roller and measure it in centimeters, and then you write it on the line. The next step it talks about is make observations about the sepals. And if you remember, the sepals are just there to protect the flower as it begins to grow and open up. So on the lily, our example today, the sepals and the petals are actually, they work together. They're the same object. And also we might have tulips the day of the lesson and daffodils, and it's the same way. The sepals and the petals are the same unit. The next step is to move into the male reproductive part of the flower. So that is the stamen. So you're going to remove the entire part of the stamen, including the filament, which is this bottom part, and the anther. So remove all of those. So I took all of the stamens off of the flower, and once again you are going to take the stamen and you're going to measure it. You only have to measure one of them, write it on the line, and then make different observations. So think about how many are there and how are they shaped, what are they used for. And then in the box you're going to draw a picture of what you saw using color, and then make sure you label the stamen, the anther, and the filament, all three parts. Okay, then you're going to move on to the female part of the flower, which is the pistil. So for the pistil, once again, you're going to measure the entire part of it. So I am going to, this is the part where you would come up to me and I would cut it for you because the razor blade's very sharp. So you would just cut it off from the stem. So once again, label all three parts of the pistil. Measure it with the ruler in centimeters. And then in the box on your lab sheet, you need to label and draw the picture. Now this last part, which I think we should have time to do, is kind of interesting to see, especially if we use the microscope to take a look at it. We're actually going to cut the pistil in half, and we are going to observe the small compartments inside of the pistil that contain the ovules, which contain the seeds of the plant. So all you're going to do on this step here is take the razor blade and give it a little slice. And with like the naked eye, you can see that there are three compartments inside of this flower, but we're going to actually put it underneath the microscope and dye it with a little bit of iodine so we can actually see the ovules inside, which contain the seeds. Then after that, the last part of the lab is to answer the 5 questions on the last sheet here using complete sentences. You are going to have to use the stuff that you learned in the lab for these questions, so make sure you have everything filled out correctly. So I guess the key here with all the pictures, just make sure you label all of them.
I'm going to zoom in on that.
Okay. Make sure you label all of them, make sure you color them, make sure you're making nice observations and answering all the questions on the last page completely. And like every lab, clean up your space when you're done. It's pretty easy cleanup. All the flower parts can just go into the trash can.